Hello, I'm Damien Fair. I'm a cognitive neuroscientist and co-director of the Masonic Institute of the Developing Brain. I'm also a professor in the Institute of Child Development in the College of Education and Human Development, and professor in the Department of Pediatrics in the medical school at the University of Minnesota. I'm glad to be here today to answer your questions.
Dr. Fair, thank you so much for accepting our invitation and having this interview format with us.
What research question do you ask in your research? And why has it received scientific attention? What are the grander issues that the field is grappling with? And how does this research question fit in? What would we know if we had already found the answers to these questions?
Well, my career has been devoted to studying the developing brain. We use non-invasive techniques like MRI to understand and characterize fundamental principles of brain organization, structure, and function. This work goes from infancy through young adolescence to young adulthood. We also examine atypical brain development to characterize mental health disorders like ADHD and autism spectrum disorders. I'd say that my philosophy studying brain development very much focuses on avoiding getting stuck in a box. So what I mean by that is that the brain is complex. Humans are complex. Disorders are complex. Yet we often try to explain the brain and its associated typical or atypical behavior, or even its treatment or interventions or strategies, in terms of simple models or concepts that are specific to our lane of research. So if you're a molecular neuroscientist, you'll try to explain some complex behavior as a result of receptors on specific populations of neurons or protein-protein interactions in support cells. If you're a systems or cognitive neuroscientist, you'll try to explain behavior in terms of level of brain activity in a specific part of the brain, or thickness or the amount of gray matter in the cortical ribbon using techniques like MRI. Now, in this scenario, because my lane is obviously the best, we tend to ignore or at minimum devalue the information from other fields. So what we have been trying to do is take a holistic approach to how to study the brain. We've identified what we've called several bottlenecks to progress and have been pursuing them in parallel in hopes of accelerating true discoveries. Some of these are quite boring. For example, one bottleneck relates to methods. We do a lot of methods development. The measurements we use for non-invasive imaging in humans are often very crude. They are noisy in terms of the data and the statistics, and this noise can lead to misleading findings that throw the field off. Unfortunately, this is often ignored, not necessarily due to maliciousness or anything like that, but simply because it's not as highly valued as it should be. So we continue to work on methods to increase the ability to get clean data that can be utilized to characterize individuals. Another bottleneck relates to early brain development. So the majority of the making of a brain occurs well before we are born, and most of the major structural aspects of development are complete shortly after we can walk. And so it's hard to understand, complex behavioral phenomena later in life without understanding the trajectory from which that brain came from. So we also focus on early development. The third bottleneck relates to what we've called the heterogeneity problem, or the assumption that when we try to relate the phenomena in the brain to complex behavior like attention or a psychopathology like ADHD, that there exists only one mechanism which occurs for that phenomena. there are likely multiple mechanisms. And so much of our work is going to have a hard time breaking through until we can characterize the variance in how folks perform tasks throughout the population. As I noted before, another leg of the stool relates to bridging molecular work and work in preclinical models. That allows us to measure the direct biology more directly with work that we do with non-invasive imaging like MRI. It's important that our work is grounded in the biology.
I have two questions for you. What is the state of the art, and how is your research novel?
Well, the brain is complex. It has over 80 billion interconnected units or neurons, and they are all connected. To simplify the science, historically we've often focused or think about how the brain works, region by region or neuron by neuron. But in a complex system, every neuron, every region is part of this complex system, and it's the system that drives function, not any given specific entity. In addition, neuroimaging, looking at connectivity from region to region, is very daunting. You need some way to kind of take this massive amount of information and condense it. And that is where graph theory comes in. It's a technique that we use in our work. Well, graph theory by its nature is relatively simple. You take a collection of nodes, and nodes can be anything from people to cities to web pages, and you link them by some line or edge, so that friends between people, roads between cities, links, or between web pages. In our case, nodes are our neurons or brain regions, and the links between them are the anatomical or functional interactions or connections. Now, graph theory has been used to study all forms of complex systems, from networks on the internet to interactions on committees and subcommittees in Congress to protein-protein interactions in yeast. And in many complex systems, including the brain, you can see an organization of those nodes and networks that is neither purely random, nor is it obviously regular, meaning A connects to B, connects to C, connects to D, right? But rather there's some non-specified random structure or non-random structure in the system. So graph theory tries to quantify that specific structure of the whole system, and then we identify, well, what does that structure and that those parameters mean to the functioning of the system. In our work, we've used this technique to identify what appear to be a relatively limited number of large-scale networks in the brain, somewhere between 15 and 20, that have very unique functions in primary sensory and motor systems and complex behaviors like learning, memory, and attention. We've also been able to see these systems early in development, as early as infancy, and highlight their relationships to psychopathologies like ADHD and autism.
What method or methods do you use, and why are they relevant for your research? What can we learn from these methods that other methods may not answer?
So this is a great question. So non-invasive imaging is an indirect measure of the neurobiology, meaning if I take an MRI and I peer inside the brain, it's giving me a picture of what's inside the brain, but not at a scale where we can really clarify the specific biology that underlies it. Because the resolution is low, any patch or pixel that we look at with non-invasive imaging has an infinite number of seemingly an infinite number of biological phenomena underneath it, contributing to the signals that we're looking at. In animal models, when studying complex behaviors, we often, on the other hand, right, we often rely on relatively large assumptions with regard to how those behaviors relate to the human behavior. But at the same time, animal models are the only way often to truly understand the detailed neurobiology. So We use non-invasive imaging like MRI to bridge that divide. It often can be measured and utilized in both animal models and human models, so it's the exact same measurement, one of the few. Now, the first step in making this bridge is to show some of that same network phenomena exists in other species as it does in humans. So in a few papers now we have done just that, highlighting, using the same technique like graph theory and network structure that is and shown that it's comparable from mouse models to non-human primates, and the same in humans as well. That the overall network structure seems to be remarkably similar in non-human primates as it does in humans. So one result to this end that I think is fairly eye-popping, at least for someone like myself in the field, it comes out of a lab from UC Davis, from David Ameral's lab, where we had a student that we co-mentored. His name was David Grayson. Now, using this technique called DREADS, which allows you to deactivate different parts of the brain, he was able to deactivate a brain region while in non-human primates while they were in the scanner, in the MRI scanner. In this case, the brain region is called the amygdala. Now, surprisingly, the connectivity from the amygdala was reduced, but more surprisingly was that the parts of the brain that were very topologically distant from the amygdala, meaning very distantly connected, meaning not directly connected, like the motor cortex, also changed. So in this case, the connectivity actually went up instead of going down when looking at the amygdala connectivity with neuroimaging. Now, at first, this might seem, at first blush, it may seem very pedestrian. But what this means is relatively profound with regard to how we interpret brain imaging that we see with MRI, and what we see with non-invasive imaging. Often when we do functional scanning and see a brain region light up, right, we assume that there must be some change in the underlying biology that's right underneath the thing that we saw with the activation. That the behavior or the change that we're seeing, or the disorder that we're looking at, is associated directly with that activity. But what this finding shows is that with non-invasive imaging, that doesn't have to be true, and in fact is most likely often untrue. That the true biological change could be very distant to what you're seeing, and cause some modified activation, but show up and be visualized someplace else. And so how we think about what we see with non-invasive neuroimaging like fMRI needs to be really well thought out to make sure we're truly interpreting the data in the right way.
Describe one or two of your most relevant research experiments. Questions, hypotheses, methods, results. What do we now know? What questions does this work answer? What new frontiers do these findings raise? Identify some key next steps that these research findings raise.
Well, there are several discoveries over the last decade that we've made, and half from myself and collaborators that I believe have pushed the field forward. What we learned from a 10,000-foot view is that the brain is organized in this relatively small number of correlated distributed brain networks. This basic principle predicted in a few classic papers, but we and others clearly demonstrated in a series of publications. But the principle is now well described, and it's often the framework by which most of cognitive neuroscience work is often contextualized, particularly in our studies of brain development. Now, back in 2014, we highlighted and characterized how our measurements can act like a functional fingerprint of the brain. Understanding how a how variable brain function is from person to person has been quite important for other discoveries related to what we call the heterogeneity problem. So the heterogeneity problem describes how the same behavior or mental health disorder, like again, like ADHD, can be related to very different brain mechanisms depending on who you are. In other words, there is not one cause of mental health disorder for something like ADHD, but likely several. In other words, it's more than one disorder. Because this phenomena is often neglected in our research, new discoveries with regard to causes and treatments have been slow. The hope is that is to now use this information to more personalize our approach to brain health. The other discovery that has grown in recent years is related to what we call the developmental programming. I kind of hinted at this a little bit earlier. This phenomena relates or highlights that many of our mental health disorders that we see appear in childhood and adolescence likely have their origins much earlier, even as early as during pregnancy. We have highlighted in humans that diet, particularly the Western style diet, things like inflammation, maternal stress, can have very long-term effects and long-term outcomes across development. The importance of this work, I believe, is that it provides very specific targets for applied and basic research to accelerate our understanding of developmental psychopathologies and ability to generate novel therapies and interventions, and to also do so at the right time for in particular for developmental mental health disorders. For example, testing prevention strategies early in development is a growing focus of the work that we're doing. We also believe that the concepts that have been borne out of studying brain and behavioral heterogeneity in the context of mental health disorders is likely going to touch other areas of our society as well, potentially assisting in improving efficiencies in education and rethinking policies geared towards this one-size-fits-all. The next steps are more about how we pull this all together to make a real impact, and that is what we have been doing at my new institute, the Masonic Institute of the Developing Brain.
What are the translational implications of your work and the work that you're doing at the Masonic Institute of the Developing Brain? And how does this work contribute to the betterment of society and humankind?
So I'm glad I get to talk about this. I'm just so excited to be the co-director of the Masonic Institute of the Developing Brain. The Institute's mission is to foster healthy brain function across the lifespan, translate this research into clinical management, community service, and effective policy. The gist of this vision is that we need to break down the silos of expertise if we are going to make progress. Developmental neuroscience, developmental psychology, non-invasive imaging, the clinical practice, computing and engineering, community engagement and outreach, particularly in underrepresented populations, the tech transfers, IP, business leaders, policymakers, educators. We need to, because of the way our structure is mostly built, a lot of these different disciplines usually do the work in a silo. And our purpose is to start building information bridges such that the impact of all of these individual entities are greater than this, are greater with the sum of their, with the sum of their individual parts. Now, the first thing that we think that we need to make this work is that you need to have all of the expertise and unique units to make it go. In other words, the stool cannot stand without all of the legs. And we are bringing together all of those legs here at the University of Minnesota. The second thing you need is energy, motivation, excitement, drive, and kind of the will to make it go. And the bridges that we're trying to build here won't just build themselves. So that needs to come not only from leaders at the university, but also from the support of our community, our donors, and many, many others. So we are working to build those commitments as well. We're lucky that we get to be located in a beautiful new building along the Mississippi River, and it will encompass several collaborating units across campus, including all of child and mental health clinics from psychiatry and pediatrics. The Institute of Child Development is world renowned. The Institute of Community Integration, the CMRR, which is the world renowned imaging center, and the Center for Neurobehavioral Development, which is the seed effort that was used to start a lot of what we're doing today. So we're going for it. There are indeed many, many new projects being worked on. We're hoping it will serve as a model to accelerate the science and put it in the hands of the community, and vice versa. Getting the community involved with the science to make sure they are part of what we're doing and the vision and the trajectory as well. So I'm very much excited about all that.
How did you go into science? When? What age? How did you discover your passion to spend a life in science?
So I grew up in Winona, Minnesota. It's the area where I grew up is relatively rural, not crazy diverse or anything like that. But I will say there were some systems put in place that sparked my trajectory toward doing what I do now. At the time, there was a heavy investment by Bob Kierlin, who was the founder of Fastenal and the State Center from Winona. And he was building a cutting edge computer access and scientific access with what was called the Minnesota Academy of Math and Science. It allowed folks like myself and others that would otherwise not have any chance at getting a peek at what some of the more cutting edge science was like and what types of things you can do. And it was amazing. I was one of the first classes at the time, and it just opened my eyes and said, hey, this is exciting. And I was like, yeah, this is what I want to do. Now it seems small, but if it was not there, who knows what direction I would have gone for my career. For a lot of folks, particularly in communities of color, that type of accessibility, that ability to see the possibilities is not always there. I got lucky, to be honest, and I was lucky enough to be in the right place at the right time in Winona. Now, I've always been interested in the brain, but it wasn't really until I was a physician associate in this physician associate program at Yale University, training and practicing in neurology, that it really clicked in that this is what I wanted to do for my career. I practiced at Yale New Haven Hospital for 2 years in the neurology department. I worked in cerebral vascular disease or stroke with the doctor named Larry Brass. I had my own clinic. I did consults on the wards. I did acute work in the emergency room. But I also did a lot of teaching to the PA students and medical students. With regard to MRI, when I was in practice at Yale New Haven Hospital, I worked on many really, I looked at one really silly experiment looking at the visual cortex in the brain using fMRI. With my friend and colleague, Dave Wiseman, who's now a resident, who was a resident at the time, but is now a neurologist in Philadelphia. We were encouraged by a physician there, Dr. Bennett Shaywitz, who gave us a little bit of resources to conduct the experiment with a resident expert, Todd Constable, who ran the MRI center there. Now, the experiment didn't go anywhere, but my eyes really awakened because it was just amazing that I could non-invasively peer into the brain of a person and characterize how it was functioning without actually touching it. Right? It amazed me. So I was hooked, And I knew that I needed to get my neuroscience PhD and study brain function with this amazing tool. So that's what I did. And that's when I ended up going to Washington University to do my graduate school. Now, it was after my PhD that I decided to determine if the techniques we were using to study development and what we were learning about brain maturation could assist in the pursuit of improving developmental mental health disorders. And here I am today, and trying to make a difference in even new ways.
What challenges did you face that propelled your scientific success? How did you overcome them?
So I think I'm going to answer this question by describing some of the maybe not my own failures per se. I mean, although I would say that it's maybe related, but some of the failures of the field and in science in general that I think is necessary to maximize the effectiveness of what we do. So the failure that I like to always talk about, at least the challenge, is it relates to variability. Okay, variability in background, variability in culture, variability in ideas. It's one of the driving principles of most of what we know in science. It drives improvements and increases fitness in an ever-changing environment, right? Variance, variability. It has been the driving factor of discovery in advance of nearly every civilization, where some outlier, oftentimes of minority status, in one way or another, pushed our society in a way that no one could have ever imagined. Yet in our space, in the sciences, despite our understanding of this to be true, we just don't value and place a value on it that's proportional to its worth. We talk about it because we know we should. We'll make adjustments to live up to those words, but we won't truly embrace it and value it proportionally to the importance in discovery and advancement. That will only change when the system itself becomes dehomogenized. So this is doing whatever it takes to consist in this process. We're trying to build inroads and trust in the community, and making sure that every kid knows that he can be a scientist too if they want, and kind of maximizing this diversity in the work that we do, the diversity that surround us when we're doing our science. I think will have tremendous impact on how good that science progresses, and how valuable it is to our society. And I'll end there. I thank you very much for having me provide this very unique talk and forum. I hope you enjoyed it. I'm happy to take any questions from folks at any time, in any forum. Thank you.